Today I'm going to talk to you another important topic, and that is the enzyme. We have already gone through in detail of this topic. Before this topic, I told you that the topic of protein and topic of the vitamins was also very important as far as university weightage is concerned. The table of specification I gave to you, a better look to that table of specification. In that table, the vitamin topic got the maximum weightage, and 14 and half marks paper will be from that vitamin topic. Similarly, the enzyme is also a very important topic as far as the weightage is concerned, and there are equal marks, about 11 to 12 marks paper, which is being set from this portion. And this portion, like the vitamin, again is very easy to understand. There is nothing difficult. If you look on the syllabus the university has given to you regarding the enzyme, that's very simple. There is a nomenclature, classification, nomenclature, very easy. Then there is certain properties of the enzyme, and then some definitions are there. That is the coenzyme, cofactor, and the similarly enzymes, allosteric enzymes. These are the definition type of things, and definitely along with that their biological significance we have to see. The other important is the factor affecting enzyme activity. So we will be dealing what type of factors they affect and the activity of the enzyme. Then enzyme inhibitors, they try to stop the reaction of the enzymes, and their importance. The inhibitors, different type classification. They are competitive and non-competitive inhibitors. You know that. And there is a mechanism regarding mechanism of action. We go through the Michaelis-Menten and equation, and similarly Wiener-Weber-Burke equation. When you was dealing this, some of the students, because you are new in the medical college first year, some of the students they try to ask the derivation. So there is in the syllabus it is written that its derivation is not required. According to, there will be no question that the paper will be set for the derivation of that. But it is only you should know what is the Michaelis-Menten equation, and similarly the Burke equation, and what is the significance of these regarding the enzyme activity. So there is nothing regarding the derivation of this. Then another very important portion of that is the enzyme is the use of enzymes in the medicine, therapeutic uses, and their diagnostic application. So that is very important, and this is all regarding your syllabus set for the enzyme. So now coming back to the point, enzymes, if we try to define the enzymes, how we will define it? The definition, how we will define the enzymes. So we can say these are the protein catalysts, and they are protein in nature, and when they increases the rate of reaction without being changed in overall process. When they are as a catalyst, definitely the catalysts are those they they increase the speed, they increase the velocity of the reaction, and they are not themselves, there is no change in their own configuration, and they just increase, they bring change in the overall process. And these enzymes, so they direct all the metabolic events. Regarding how to name these enzymes, whenever there is a name of A's, A's at the end, that will let you know that this is the enzyme. For example, there is a I give you an example, glucosidase. For example, it is glucosidase. Now, it is the A's at the end. So it is an enzyme. It is specific. At specific place, it is an enzyme. Similarly, urease, urease, urease. It is the word A's at the end. Look, urease or urease, you will call it. And whenever it is A's at the end, that show that this is an enzyme. This is how the enzymes are given the recommended names. There are certain enzymes in the gas which are not, there is no A's at the end, but still they work as the enzyme. That is, for example, trypsin, pepsin. They also act as enzyme, but not the name of A's is not given at the end. As far as I'm talking to you regarding the classification. After going through how to name this, then is the classification. Very easily, the enzymes been divided. There are 3 ways of classification of the enzymes, but the most recommended is that they are of 6 types of enzymes. And what are those 6 classes? One is the oxidoreductase. This is second, transferase. I'm saying it is word A's at the end, so it is transferase. Then is the hydrolase. It is then is the lyase or lyases and isomerases. These are 5. And then is the 6th one is the ligases. So it's here. It is ligase. Now, there is sometimes question in the university that how you will classify the enzymes, give the names with the example. So this diagram is given in your Lippincott book, and from where I have taken it, and this is very good to understand. Let's say one by one we go on talking. That is the oxidoreductase. And the example here given is when the lactate is converted into pyruvate. You will study this in the metabolic pathway. When the lactate is converted into the pyruvate, there this oxidoreductase is come into an action, and this catalyzes the oxidation reduction reaction. This is example. This is oxidation reduction reaction. It is catalyzing here, and example is the lactate to pyruvate. There are many other examples, but just to understand, one example is being mentioned here. Second is the second class of the enzyme. We are classifying enzyme with example. Transferases. It catalyzes the transfer of carbon hydrogen phosphate containing group. for example, all those groups where the carbon, nitrogen, or phosphate present, these transferases, they come into an action. They are important. And for example, serine, when converted, individual amino acid, talking serine converted to glycine, at that place, there's this transferases enzyme is mentioned here. This enzyme will come into an action. This is a transferases, and it will transfer. That is, the group is being transferred here. You can see it is mentioned red here, CH2 being transferred here, like that, and this is transferases. Then hydrolases, that is the catalyze the cleavage of the bond by addition of water. Very nice to understand and very easy to understand. There is this is the cleavage of the bond by addition of water. Here the bond is being, it is mentioned here, he is showing very rightly this water being here, and that bond is cleaved, and there is then this product is being formed. The other class I'm telling you the classification of the enzymes with example. Lyases, it catalyzes cleavage of carbon-carbon bond, carbon-sulfur bond, and certain carbon-nitrogen bond. Certain amino acids, certain products, substrates. When we were talking of the enzyme reaction, we have to talk of the substrate. And the substrate is composed of carbon-carbon bond or carbon-sulfur bond or the carbon-hydrogen bond. So lyases, they will break down, cleavage will be occurring here as indicated here, where pyruvate is converted into acetaldehyde. Carbon dioxide is being removed here, the bond is being broken here. It is being mentioned red here that bond is, that is cleavage of bond taking place here, and this carbon dioxide is then being formed, and it is here you can see. Then is the isomerases. Word itself is very clearly that catalyzes the recommendation of that is the cementation of this special or the geometric isomers, such as methyl CoA to succinyl CoA. These are all reactions which are in the metabolic pathway. So what this is indicating that these enzymes, they help the metabolic pathway to proceed. They speed up the process of the metabolic pathway due to their catalytic activity, because they are catalytic, they have got the catalytic activity also. And you understand what is catalytic activity? That the catalyst, they enhance the speed of the reaction, they enhance the velocity of the reaction, and without having any conformational change in their own structure. Then the sixth one is the ligases, and here that is it catalyzes formation of the bonds between carbon and oxygen, sulfur, and nitrogen compound, and this is being indicated here that here is going. This is the example is the pyruvate to oxaloacetate. Now once again, repeating that there are 6 classes of the enzyme. That is that is one is the oxidoreductase, then is the transferases, then is the hydrolases, then is the lyases, isomerases, and ligases. The best way to remember, you all very good in remembering all that, but you just take the first word of OTH. You remember OTH, and second is LIL. That will you will always remember by keeping these first word in your mind. You will be in position to write down the next one. So there are certain now. I have discussed with you the enzymes. And I was talking to you that these are 6 classes, but there are certain potentially confusing enzyme nomenclature. This is here, but no need to be confused. For example, that synthetase, for its it requires ATP, but the similar type of name, little different, is the synthase, no ATP required. So these are sometimes creating some confusion. So there is no need to be confused. That is the synthetases that requires ATP, whereas synthase, it is not required any ATP. Similarly, phosphatase, tase that is use water to remove phosphoryl group. Now I am talking this. It is use water to remove, that is phosphoryl group. Whereas the phosphorylase, difference is phosphatase, phosphorylase use phosphate to break a bond and generate there is a product, then it generates the product. Similarly, dehydrogenase, it is an electron acceptor redox potential. Oxidase, oxygen is the acceptor and oxygen atom are not incorporated into the substrate. Whereas oxygenases, one or both oxygen atom are incorporated. Here, number 4 and 5 I am talking. There were, in the case of oxidase, no, and oxygen atoms are not incorporated into the substrate. Whereas in oxygenases, one or both oxygen atoms, they are incorporated. So these were some enzymes that may be creating some confusion. Same thing is mentioned here in the book, that is the potentially confusing enzyme nomenclature. I have talked with you in detail in my previous one, and so this is the same thing what I broken form I have told you, that is how to present it. Then now how these enzymes, when I talking now coming to the properties, after this nomenclature, their classification, then is the properties of enzyme. As I told you, they are the catalyst that increases the velocity of the chemical reactions, and they themselves are not consumed during the reaction. There are some RNAs. They can also act like enzyme. And these RNAs with the catalytic activities are called isozymes. Maybe definition of isozyme, you can give example of RNA. We have the catalytic activity, and they are called the ribozymes. As far as their how they act, the enzyme, this diagram is very nicely in telling the what is the mechanism of action of the enzyme, how these enzymes come into action. This is a substrate. For the action of the enzyme, the presence of substrate is very essential. Substrate, different type of substrate will be there, and you can see this is substrate, blue color, and here is the enzyme. This will come, and this is the active site on the where the reaction will be taking place on the enzyme, on the substrate molecule. There is a active site. This will come into action. They will unite at the place of the active site. So this active site is the place where enzyme and substrate, they come close to each other, and in this way they form ES complex, enzyme substrate complex. And this complex is then further processing according to its reaction velocity, reaction, action. The enzymes, they have, I told you, the catalytic activity, and you see, they enhance. And when they enhance the speed, it may increase the speed up to 10 raised power 2 to 10 raised power 8 times faster than the when the enzyme is not available. So enzyme enhance the speed of the reaction, and the limit of that enhancement is here, that is 10 raised power 2 to 10 raised power 8 also. And there is one definition which is required here, that is the turnover number. Turnover number. What is this? The number of molecules of substrate converted to products. ये जो जब इसके बाद product बनेगा, the number of molecules of substrate converted to product by per enzyme molecule in a second, that is per second, is called the turnover number. So this is the definition. When you will talking of the property, you sometime may be asked the turnover number. So this is the turnover number of that. Now the enzymes, they are specific. When talking of this diagram is very clearly indicating, because in the cell it is not that everywhere the catalytic reaction or the enzymatic reactions are taking place, but in the cell there are different locations for the different enzymes, and this is how a systematic mechanism of reaction, enzymatic reaction, taking place. So we can say that the enzymes are specific. So if you look here, this is a cell structure, and in the cytosol, what type of enzymatic reaction, that is, for example, is the glycolysis, where there will be, take here, it will be different place where these enzymes will be coming, and specific enzyme taking place here. Then is the pyruvate kinase, the fatty acid synthesis is also here, it is specific. And if you see the mitochondria, the glycolysis is taking place in the cytosol, in the cell, is a separate place, whereas the TCA cycle, the next product of the glycolysis, where pyruvate acid is formed, and that is ultimately converted into tricarboxylic acid, and that is through TCA cycle, that is taking place in mitochondria. And all the enzymatic reaction here, they are taking place in the mitospecific. Nucleus, only DNA and RNA, there is synthesis, and all the enzymes which are located, you needed, they are being taking interaction here, and is the lysosomes here, that is the degeneration of complex molecules. So what I mean to say is, this is the intracellular location of important biomolecule pathways. Intracellular, this is intracellular location of important biochemical pathway. It is not that at only one place every type of reaction is being taking place, but it is separately, at separate places, separate enzymes, they are coming into action. Now, next is that there are regarding properties, there are certain definitions which are very important, and these definitions are the holoenzymes, the apoenzymes, the cofactors, and the coenzymes. These four definitions you must know, and this is given in the book, but I have made these separate headings, that the term holoenzymes refer to an active enzyme with its non-protein compound. When it is non-protein compound, because there is a protein, basically the enzymes are protein, but if it has got the non-protein portion, that will be known as the hollow enzyme, whereas the enzyme with its here, whereas the enzyme without its non-protein moiety is termed as the apoenzyme and is inactive. This is apoenzyme. This is here, it is apoenzyme, and if that non-protein portion is metal ion, that is zinc or iron, it will be called as cofactor. Very easy. You can make such a statement yourself. If it is a small organic group, when it was metal, zinc or iron, it was cofactor, and when there was non-protein that moiety, that was the apoenzyme. And if it is an organic molecule, one portion is not enzymatic or protein, and attached with that is an organic molecule, it is termed as the coenzyme. Coenzymes that only transitionally associated with the enzymes are called substrate. So similarly, it has separately given, this is given in your book. So these definitions you must know, and because sometime in the university question there is a definition out there. Now quickly going through the activity of the enzyme. Now the factors, let's say the factor affecting the enzyme activity. First, the enzymes, there are different types of enzymes, their classification, and now another important issue is there are certain factors which affects the enzyme activity. You see, at a certain temperature, these enzymes, they work, and their velocity increase, but then a peak is reached. After that temperature, the enzymes, you know, they are protein in nature, very rightly, and you also know that protein, they are denatured at high temperature. So because they are protein, they will be denatured, and this is what this definition, these four lines are indicating, that the optimum temperature for most human enzyme, optimum temperature for their good activity, is between 36 and 40 degrees centigrade. At this, up to this temperature, the enzyme, that is 35 degree temperature and 45, this is the optimum temperature, the activity will be going on nicely. Human enzyme starts to denature at temperature above 40. When temperature will be more than 40 degrees, there is denaturation of the enzyme. And why denaturation will be taking place? Because they are protein in nature. But thermophilic bacteria found in the hot springs have optimum temperature of 70 degrees centigrade. This is different. So what I wanted to tell you, that this factor of temperature, it affects the reaction speed and reaction. Now this is diagrammatic presentation also, that is the effect of an enzyme catalyzer. You see, it will go to a maximum and then will start coming down. Maximum will be going up to 40 and then will be dropping. This is here, and after the optimum, then the reaction, this red indicating the denaturation then starts, and then there is a heat inactive, makes the enzyme inactive, and this is what is the effect of temperature on the activity of the enzyme. Similarly, there is the effect of pH. You know there is certain up to certain pH enzymes they work very good, but if the pH is increased, so denaturation of the enzyme will take place, and the typical example and very good example is in the stomach, the pepsin, the trypsin, and the alkaline phosphatase is example. It is given here in the diagram, and in your book it is also being mentioned here. This is quickly I'm telling you some important factors which are affecting the enzyme activity. Another very important from examination point of view is the Michaelis-Menten equation, and you have not to drive it, but you have to just write it down. You should know what is the Michaelis-Menten equation and what's its application. And here I have shown you the Michaelis-Menten equation. You must know what is Michaelis-Menten equation, as it is regarding with the reaction velocity, and V initial reaction velocity, V maximum, max means maximum velocity that is reached, and Km the Michaelis constant, it is here mentioned, and S is the substrate. So this Michaelis-Menten equation you should know. There are certain assumptions they are made in driving the Michaelis-Menten equation. Well, we are not going for the